My guest here for the next 40 minutes is Michael Sauer from BOKU, the University of Natural Resources and Life Sciences in Austria, in Vienna. So, yeah, are you enjoying a nice, crisp Vienna morning at where you are today?
I do. Actually, we had a very nice week, but today is the first day of rain. But
okay.
That's okay. We're all in it anyhow.
I just had this image of you waking up and going to a fantastic Austrian coffee house and having a nice warm coffee in the morning as you look at the mountains. Is it not quite like this?
It's not quite like this. From Vienna, you wouldn't see the mountains. And if I have an appointment at 8:20, I would not go to the coffee house before.
Well, then my fantasy has been ruined, but never mind. But thank you for joining me on International Microorganism Day. And we're going to have a bit of a chat about lactic acid bacteria and yeast.
Yes.
So I guess, this year more than any, people have familiarized themselves, whether knowingly or unbeknowingly, with cultures of microbes to make sourdough or to make homebrew or beer or so on. And so it's quite interesting for us to explore these kinds of bacteria and yeasts. And just before we delve into some questions, I just wanted to point out to anyone on the call that we have some educational resources on sourdough and how to make your own sourdough. So I'm just going to briefly point people in the direction. Ooh, my mouse seems to have stuck. This is always classic. Of the International Microorganism Day website, because we have some wonderful education resources. So if my computer will allow me, I can prompt us with some lovely illustrations.
Absolutely.
Yes. Can share this screen. Cool. Okay. Can you see that? Is that visible for you, Michael? So yeah, if you go to the International Microorganism Day website, come to the educational resources section, you can find a fully complete infographic on how to make sourdough bread. And as you'll see from these instructions, a lot of it involves setting up a starter culture, which you have to feed and maintain throughout the process. So this is where I come to you, Michael. With lactic acid bacteria, firstly, could you give me a bit of an indication as to what are these bacteria? Why do they have this kind of odd name? And where do they normally live?
Okay. Lactic acid bacteria live everywhere, essentially, but particularly in environments that are very rich. So you would find them, for example, on plants. So if you take some plants from the environment, you would find lactic acid bacteria, but they also live in and on animals. So in your body, for example, in your mouth, in your intestine, wherever you would look, you would find lactic acid bacteria. And they are named like this because their main product, or the product that is very easily visible, is lactic acid, which is a very pleasant tasting weak acid that we use a lot in.
So this is the same acid that develops in your body when you exercise?
Exactly. That's
yeah,
the same
okay.
Reason. And it develops actually for the same reason. The point is that it develops in our bodies when you need a lot of energy. So when you go running, for example. And to get this energy, usually what we do is we burn sugar. You really can imagine it like a fire. So you use oxygen to burn sugar, and that energy you can use for running. But if you don't have enough oxygen because you can't breathe so much, let's say.
Yeah, okay.
You have to find other ways. And then you would produce lactic acid. And that's exactly what lactic acid bacteria do. When they are happy if there's no oxygen. And that's when they produce lactic acid.
Okay. So then if we just, if I just point us back in the direction of the instructions for the sourdough bread, one of the things I noticed was there's no particular stage in which you're particularly putting a sample of lactic acid bacteria into this culture. So is the idea that they're just so ubiquitous from my touching of stuff that they will get into this culture from my skin and then start becoming a live culture that we can then use to make bread.
From the grain. So
Yeah.
when you take the grain from the field and you would grind it, but then still lactic acid bacteria are on it and they will be happy to grow. Actually, I brought one starter culture to show you. It gets a little bit out of control right now. So,
oh, wow. Let me just stop sharing so we can see you in more detail. Okay, yes.
So this is how far it was filled yesterday night. So with flour and water, essentially. So, and the culture that I have in my fridge, actually, and I'm feeding. And this is what happened overnight, and there's not a lot of space left and
oh, wow.
control. But you can see the air bubbles inside.
Yeah. So what gases are those bubbles made from?
This is carbon dioxide. It's
okay.
Exactly what we do. So when we breathe, we produce carbon dioxide, and this is what also lactic acid bacteria, or in this case also yeasts, do. And this is what we want, because that makes the bread so nice with all these holes inside.
So, I mean, I think you've answered my next question, which was going to be, how do they give bread its distinctive taste, or sourdough the distinctive taste? And so it's the presence of this extra acid that is the slight tingle on the tongue, or is it another thing?
Actually, it's a combination of things. So one thing is definitely the acid. So if you have a good rye bread, then you will feel that it's slightly sour tasting, and this distinguishes it from other breads. And that is definitely the action of lactic acid bacteria. But the point is, these are living organisms. So they take up stuff from, let's say, the dough. So like some amino acids, some other things, and that will change the taste. And they produce not only lactic acid, they also produce other things, aromatic compounds, and also that will add to the taste. And so this is why, depending on which bacteria you have in your culture, the bread will be different, even if you take the same, let's say, flour.
Okay, so if we have people in different parts of the world following the same instructions, due to their local bacteria populations and the microbes that they end up growing in their starter culture, their breads could be slightly different in flavor and taste.
It will be slightly different. And you can even test it at home if you want to, because like it's kind of a natural selection. So you take your flour exactly as it says in these instructions, and you do it. And then, of course, let's say the ones that are most happy with the environment that you create will prevail. They will dominate your culture. And so, like us, we feel good at different temperatures. I like hot weather, for example. Other people might like cold weather. And so there are bacteria that are healthier at warmer temperatures or at lower temperatures. So if you prepare your sourdough and you incubate it, let's say at the coldest place in your apartment or at the warmest place, you will have a different culture in the end, and it will taste different.
Okay. So then it's, whatever microbes we have and what they produce that help to change, the flavors, but also the textures of the bread. Because I always find sourdough is quite tough. Is that due to sort of the protein or something behind the bacteria growth, or what's that extra element?
Yes. The point is, traditionally you would do sourdough particularly with rye flour, and this actually is a very difficult flour for baking bread, and if you just try it with yeast alone, it will not work properly. Again, there's a lot of chemistry behind, but to make this flour, for example, stick together, you need some sticky proteins that keep it together. And in order to develop that in rye, you need an acidic environment, and then it works perfectly well.
Okay, okay. So it's also the flour that would be used. So it's the combination of these microbes and the specific flour.
Exactly. If you take completely white wheat flour, you don't need sourdough. You can just do it with yeast, because that will stick perfectly well and you can bake it easily. But if you try the same thing with rye, it would not work because it's just not sticky enough.
Yeah, okay. And so in these cultures then, when the bacteria are producing acid, do you have any idea of how strong the pH, how strong the acidity gets within the culture? Is it something particularly acidic, or is it still within, you know, almost a neutral range? Do they have to do anything special to withstand the acid they're producing?
No, it is quite acidic. So I would say it like neutral would be 7.
Yeah.
And it would go down to 5 or even below 5, can depending on the fermentation that you're doing. And that does make a difference for the bacteria. And actually also that is one of the things why it works so well, because in this case, lactic acid bacteria are a little bit mean, because by acidifying their environment, they kill or inhibit their competitors that could also grow. So this is to make a side step, for example, working when you make sauerkraut or pickles or something like this, that is the same principle. So they have to have special measures to survive it themselves, of course.
So, I mean, when we're making bread, we don't just have lactic acid bacteria, we also have yeasts, right? That are helping to ferment the dough. So do the lactic acid bacteria in sourdough cause any trouble for the yeasts? Is there anything we have to worry about? Perhaps our yeast getting killed off by the competing microbes in the mixture?
Yes and no. The point is there are a lot of yeasts that do not like such an environment. So for example, the typical baker's yeast, even if it's acid tolerant, it doesn't like so much the environment in sourdough. So in sourdough, you would have different yeast species that are more adapted to low pH.
Okay. So I have a picture of yeast, which I can share just to kind of put this in a nice perspective for everyone. So it's the classic yeast. We have the classic Saccharomyces cerevisiae, which is, you know, the standard baker's yeast. And here we have some cells just sitting there on the microscope slide. But we also have then some lumps of fresh yeast and then some pellets of dry yeast. But in the case of our sourdough, we think we'd have a more complicated community of yeasts than just this standard bake. Okay. And that would hopefully allow you to ferment bread even though it's acidic. Now, yeasts don't just make bread for us, do they? They also make wonderful things like beer and wine.
Exactly.
And one of the things I think is good to ponder about is, you know, we enjoy the fact that they make alcohol. It happens to have an effect on the human brain. But why do yeasts bother to make alcohol? What's in it for them?
If we think about baker's yeast, that's a very special organism because it's extremely efficient. So it can produce alcohol very, very, very quickly and to high concentrations. So why does it bother? These questions are always very difficult to answer. And what I have to start off with is that, of course, it's usually, let's say, our idea that we interpret into the yeast. It evolved in nature to be as it is, but why it really did it, who knows? But our current idea of it is the following. The point is, it's again, you have to imagine in nature, it's always competition. So you have a lot of organisms there. And what actually each of these organisms wants to eat first is exactly like us, it's the sugar. So we like the sweet things, and so microorganisms do like the sweet things. And so the winner is the one who can eat the sugar first. And so baker's yeast now has a metabolism, so that means it can take up the sugar and get some energy out of it, that is extremely quickly. And in order to do so, what it does, it converts the sugar into ethanol. This ethanol has two effects. One is that it is quite toxic for many other organisms. So like the lactic acid bacteria are mean, also the baker's yeast is a little bit mean to their fellow yeast organisms because it kills them by making so much ethanol. The other thing is that it deprives the others from sugar because it's just quicker in eating it. And so what it does, it just eats the sugar, so the others have no sugar left. It makes ethanol, which kills the others. And when, let's say, it's essentially alone, then it can start off and eat the ethanol very slowly and grow also on ethanol. And in the end, all the sugar, let's say, will go to yeast.
Okay. So I have, again, some more pictures, which I've assembled for this moment. And here we have now a nice bottle of, I guess, some kind of wine sitting there, fermenting away with some yeast. And we can see the yeast building up here as a sediment, right? So this will be turning the sugars in our juice, I guess it could be a grape juice or some kind of sugary juice, into the alcohol.
Yeah.
And so it's kind of, as you indicated there, almost like a sort of cheat in a sense. It's trying to ferment the sugar quickest. Producing alcohol happens to be the byproduct that allows it to do this. But you just mentioned it can also consume the alcohol.
Yes.
Okay. So is there any risk then that if we leave our brewing for too long, we'll start to actually reduce the amount of alcohol that's in our brew?
Not how we do it, because in order to use alcohol, the yeast needs oxygen. If there's no oxygen, alcohol cannot be used.
Okay.
And so usually our bottles, barrels, casks, whatever we have are closed, so there's no oxygen, and that means that yeast cannot use the alcohol.
So I think I have an image. Yeah, here we go. So this is kind of how we brew beer, at least at home, maybe. So these valves here, will let out carbon dioxide or any other gases, but stop oxygen getting in.
Yes, which is very important, by the way, because the amount of carbon dioxide which is produced is very high. And actually, yeasts are very tiny, but they are very, very strong. So if you would close such a bottle tightly, the yeast would make it explode. It's actually dangerous. So if you do it at home, be careful to really let it open at one point or add a valve or something. Otherwise, it explodes.
I guess you couldn't really tell because the pressure would just keep building up inside.
No, you can't tell.
Almost invisibly until...
Exactly.
Okay. So can you tell me then what's going on here? So I guess this is an industrial brewer. And I guess we have yeast and gas coming out the top of the brewing, but they must have just taken the lid off or something, because you say ideally it would be closed so that the oxygen can't get into the brewing sample.
Yes, I'm not entirely sure which step it is. It could also be the cooking, because at one point, for example, if you do beer, then you would cook your malt, and it might be just the cooking stuff. But there are also beers which are done in open casks, because they want like also wild, let's say, microorganisms ferment their beer. That gives another very peculiar taste. Usually also lactic acid bacteria add in, and so it gets a little bit sour. What definitely is interesting is that we have very different yeasts behaving in a different way. So some of them, when they ferment, they go down. And
so this is the bottom brewing yeast brewing types.
Exactly.
Yeah.
Exactly. And others, like for ales, they would float on top. That would look like on your picture.
Yeah, because I mean, so I grew up in the United Kingdom. People make lots of ales there, so they're using the top. Yes. I guess the top brewing yeasts, and I guess is it most traditional sort of pilsner or lagers will be having bottom brewing yeasts? Exactly. Is it this way around? Okay. And what makes the difference? Why does one float and one sink? Is it just the density of the cells or something? What's making them favor different positions?
It's particularly how the cells stick together.
Okay.
So there are different possibilities how cells can stick together. And if they stick in large, let's say, chunks, they would sink down.
Yeah.
They more build fluffy, let's say, structures, which then fill even with gas, then they would float. Okay,
so it's also about not just the density or weight, but the surface of these cells and how they stick together. And will that be down to different proteins or different?
Exactly.
Yeah. Okay.
Proteins, and some of them we know, so we know exactly if a yeast has that protein, then it would stick tighter. If it has that, it would stick less, whatever. Just to tell you about explosion, so here's something that's completely out of control now.
Well, let's have a look. Oh yeah, I see. Yeah. Okay, so you've got some overproductive microbes there.
And so, of course, it's not closed, so it can come out. It will get worse until the end of our process.
So, Okay, so we need to make sure that we don't overly seal any brewing, so we don't explode our kitchen, garage, or shed. And we need to make sure we have the right yeast, or at least we've got a yeast strain that we really want to be using. Actually, I think if I just quickly share my screen again, we can perhaps see the difference between these yeasts. So I'm guessing that this one here is a kind of bottom brewing yeast because it's sitting at the bottom of the wine. And if we move across to this one, you can see the sediment here is at the bottom. So this would be another bottom brewing yeast. Although this beer does look quite ale-y, but yeah, I see. It's got some nice color to it here. Because that's the other thing is, I mean, when I was growing up in the UK, lots of pubs, lots of ale, all those beers are still live when they get them into the pub. So that the casks still have a live culture of yeast living in the beer. So they don't kill it off or anything. And so over time, the flavor can change and the alcohol content even can change. And eventually it gets to the point where it's kind of vinegary. So is that the next step in the fermentation? You've taken sugar, it's gone to ethanol, but if you then let too much oxygen in, it takes the ethanol into, well, I guess ethanoic acid or vinegar-like acid, which is where we don't want to be.
Yes, we don't want that, but that's actually another guy inside there because these acidic acid bacteria, they would consume ethanol and produce vinegar from it. So if you produce vinegar, that's the way you do it. If you want to drink your beer, that's not what you want.
Yeah.
But that's other bacteria that are also everywhere. So if you put oxygen into an ethanol solution, you would very easily get that. Yeah.
Okay. All these clever microbes doing fun things for us. So yeah, this is another thing. So I mean, this alcohol, I think we could all testify that if you have too much alcohol, it starts to feel like a poison and starts to make you feel quite ill. Is the same true for the yeasts? And if so, how have they evolved to stop themselves from being overwhelmed by the alcohol in the solution that they're growing in?
It is the same. It is the same for all organisms, also for yeast. So definitely there are concentrations which would be even deadly for yeasts, but obviously they're not producing them. Definitely there is a point when they stop to live, or when they stop to do fermentation, when the ethanol concentration gets too high for them. They have certain measures to shield themselves. So for example, by changing their membrane to make it more, let's say, rigid is not the entirely correct term, but you can think of it that the ethanol cannot enter the cell. Or they have very efficient pumping systems. So they have systems that when ethanol enters the cell, they pump it out and get rid of it. And in this respect, they are better than their competitors. So every cell has certain measures to cope with ethanol, but some are better, some are worse, and yeast is quite good. So it can produce like 20% of ethanol.
Okay, yeah. So I guess so my sort of next question is, if we're brewing beer, it might stop at say 5%. If we're brewing wine, it might go all the way up to 13 or 14%. So what's the reason that these different brewing yeasts stop at different percentages? Is it just when we stop the reaction, or is it down to their innate biology?
No, actually that depends on how much sugar you give them. Because they will convert every sugar you give them up to a certain amount into ethanol. And so for beer, we usually add the sugar that it will end up at between 4 and 5%, but which is not entirely true, because if you look at Belgian beers or Bock beers or whatever, so there are also beers that go to 13, 14%. This is then called the double or triple or quadruple.
Yeah, they're very common here in the Netherlands.
Exactly. That simply means that you add the double or triple or quadruple amount of sugar.
Oh, yeah.
And that means that you get the double, triple, or quadruple amount of alcohol in the end. And grape juice is very high in sugar. And this is why wine has much more alcohol.
Okay. Because actually, I remember reading the labels on those double, triple, quadruple beers. And if you look at the ingredients, sometimes they have glucose syrup added specifically to the brewing mixture. And I guess that would be then to up the sugar content.
Exactly. That's in my personal opinion quite cheating. You shouldn't do that.
I agree. I always look at it as a sign of someone kind of supercharging their beer against the laws of nature.
Exactly.
But, you know, if you want to get a high alcohol content in a cheap way, it's easy. Okay. But yeah, it goes against, there's the German, I guess, purity laws for beer, aren't there? And you can only have hops, water, and barley.
Exactly. And since they didn't know when they formulated the law about the yeast,
they didn't know.
It's not inside of the law, let's say.
Yeah. Because I was reading the other day about wild beers. So, I mean, you know, in most commercial beer production or commercial alcohol production, you'll be using specific strains that the brewery has decided are the one they want, right? Yeah. But in wild beers, you take your barley, your hops, and you mash it up and you produce a malty liquid. Yeah. But then you just let the natural microbes ferment beer into the juice into beer. I mean, is there any risk behind this kind of method? Because if you don't know what microbes are in your solution, could it potentially pose any risk to human life? Or are they kind of self-correcting in the sense that anything pathogenic will be killed off by these super adapted microbes for the alcoholic environment?
Yeah, I don't think that there is a true health risk that you would really produce something very poisonous with a procedure like this is quite impossible, I would say. So I think you can drink it. The risk is that it tastes awful.
Yeah.
I think the worst thing that happens is that your bowel movements will be unpleasant after drinking it, but I don't think you will die.
Okay. So, I mean, is there any knowledge on how consuming fermented food, whether it's like bread, whether it's alcoholic beverages or other things like kombucha? Actually, I have a picture of kombucha and the microbes inside it. Yeah. Is there any discussion of how these foodstuffs change our gut microbes? Because it's a very big topic these days, is what composition of microbes live in my gut? Do these food and drink products change what we expect to find in people's guts?
There is a lot of discussion about it, and I think there's not so much knowledge about it. So there are very different theories of what you have to do and how it really influences your gut. So one of the things is that what do you give to feed your gut bacteria, let's say? And so there are these certain sugars or something like this, or polymers that we humans would not be able to digest, but so they can, let's say, go quite deep in your intestine and feed your bacteria. The other thing is if these organisms can survive. So like if you think about like all these yogurts or whatever, where you have these favorable lactic acid bacteria, at least the theory behind is that when you ingest it, that they will survive in your gut. So you can positively change this environment. But the indications are that it doesn't work like this. So even if it sounds very plausible to us, and if it's a very good advertising thing, in reality, it looks quite different.
Okay. Because actually one of the talks that we had playing earlier in the live stream by Omalulu Fulganawa, who's a PhD student in the University of Huddersfield, he was looking at how gut fiber changes.
Exactly.
Yeah. So he found that, I think that was more of a effect on your gut microbes. It's not like what yogurts you're eating, but whether you had lots of fiber in your bowels to create a nice environment for certain bacteria. And that had a much bigger effect, you know, the vegetarian diet versus the classic Western diet.
Absolutely. That is what I meant, because there are constituents inside that we cannot use as humans. And so we don't digest them, but then bacteria can digest them later and be happy.
Yeah.
So let's say one experience that most of us can do is if you eat a lot of beans. Everyone knows about the effect of beans. So why?
Yeah. Okay. Yeah. Beans.
This effect. Beans have a very rare sugar inside that we cannot use. So this sugar is, you cannot find it a lot of time in nature, but beans are quite rich in it. And we cannot use this sugar. So when we eat beans, we take the proteins out, we take the other sugars out, but this sugar simply remains. But in our bowls, there are bacteria that really love this sugar, and they produce a lot of gas growing on this sugar, and this is why it has this effect. So
Okay.
you can really see by feeding them with the special sugar, you can really change their behavior.
Yeah. And I guess if you're lactose intolerant, the same is true with any dairy product. You have this, for you, that's the sugar you can't consume, like the beans, and it would make you very gassy. Okay. So something that's been chatted, you know, either consumed or shared a lot, at least in my experience recently, is being kombucha and kombucha recipes. And this is another example of yeasts and bacteria fermenting product for us.
Yeah.
And I guess the thing that people need to get their hands on is the SCOBY, which is kind of sometimes quite coveted. And it's this very strange gelatinous disc that floats at the top of the mixture. And the thing I learned, you know, the other day was that I thought SCOBY was some ancient term, some kind of, you know, primordial description of what this thing was. But no, it's just an acronym for a symbiotic culture of bacteria and yeast.
Yeah.
But here we can see some, bacteria and yeast, which I guess would be making up the SCOBY. So, you know, when we saw you mentioned earlier that the yeasts have these proteins that stick them together that make the different positions within the brewing. And I'm guessing then in this case, this symbiotic culture between these different microorganisms will be producing some kind of extracellular materials that stick them into this lump that floats in your kombucha recipe. I mean, do they do this just to kind of protect themselves or to make a nice environment for themselves? I mean, what's the reason that they bother throwing all this useful material outside of their cells?
Yes, I would guess so. Again, this is like our human view.
Yeah.
Why organisms do something.
I mean, it's we're taking our macroscopic perspective and forcing it on their tiny world, but yeah.
Yes, but I think that's the point. So they are creating an environment which they are kind of safe, and they can live, and they are not floating away, and they are also, like, a lot of time working together is very important also for microorganisms. And so it can very well be that these yeasts want something that the bacteria produce, and so the yeasts create an environment to make, let's say, the bacteria around them happy, and so the bacteria can grow there and give the yeast what they want. It could be a vitamin, it could whatever it is. I don't know if this is the case in this particularly instance, but there are a lot of like communities like this, where, let's say, one does something for the other and benefits him or herself.
Okay, and all the microbes in this community get the benefit of working together in some way.
Exactly.
Yeah, that's the fun thing that I think I've learned at FEMS over the past 2 years I've been living here, living and working here, is that it's the cooperation between the microbes in different situations, which I found so interesting.
Absolutely.
And the little communities they form, they're not even in the same domain, same domain maybe sometimes, but even that can be a bit combined. And then you have these radically different life forms finding ways to live together. I mean, they have little strange wars now and again, but they do cooperate on a huge scale.
Yes.
Right, okay. Right, I'm just going to stop my screen share because we've only got a few minutes left. But it has been fantastic chatting. I just want to make sure that everyone is fully aware of how to make their own sourdough, having learned so much about it. So like I said earlier, education resources on the International Microorganism Day website. You'll receive instructions as to how to make sourdough, and you can think about all the things we might have learned today, but there's also a bonus pack, and it's these burping bags. So these make use of yeast and its ability to ferment sugar into carbon dioxide. And you can actually test that exactly yourself. So you have a sugar solution and a non-sugar solution. You add yeast to each, and you can see slowly over this experiment, when you heat up the yeast, which ones produce a burping bag full of gas. And, you don't have to take it, don't have to take our word for it. As good scientists, you should do your own experiments, but you might expect ones with the sugar, as we learned today, to be fermenting that sugar into, in this case, ethanol and carbon dioxide.
Yeah.
Yeah.
And I can show you once more the out of control sourdough starter, because now it's really getting...
Oh, it is. So is this just because, did opening it increase its rate of fermentation, or was it already producing this? And
Just that I should have taken obviously a larger glass, or I should have used it by now to
yeah,
bake
okay.
bread.
So are you going to use this to make any bread, or?
Yes, now
Yeah.
we can use it to make bread.
Okay. So, I mean, have you now managed to replace any kind of external bakery with your own supply of sourdough?
Oh, honestly, yes. For
Yeah.
us personally, so I'm one of those people that use the staying home time in the pandemic to start their own sourdough. So this is the
Oh, wow. There's the actual bread. Looks very delicious. Because I must say, you know, when I lived in London, sourdough was everywhere. And now I live in, you know, South Holland. My access to sourdough has been much reduced, just because of the different bread culture. But I should probably make my own sourdough
Absolutely.
culture, and then I can have it all the time.
Exactly.
Okay. Well, I promise you then I will make my own sourdough, or at least have a go, because I have been missing it, and this has inspired me to make my own. Fantastic. So what are your plans today then? Are you teaching today? Is your next engagement an academic one, or?
My next engagement actually is how to improve my online presence.
Oh, okay. So you're doing a little bit of homework already, perhaps?
Exactly.
So in that case, I mean, you have a Twitter account, don't you? We can point people towards if they have any extra questions.
What?
You have a Twitter account.
Oh, I do.
Yeah. So, I mean, if people have any questions about sourdough or lactic acid bacteria, they can find you on Twitter.
I think it's Michel Wien. It's
yes, that's the one. At MICHL_WIEN.
Yes.
Fantastic.
Very happy about any questions.
Cool. And then I might contact you for tips if my sourdough goes wrong. But yeah, I see my next guest is in the waiting room. So we must sadly move on from sourdough and move into magnetotactic microorganisms, another fascinating subject. But thank you so much for joining us. Good luck with your next engagement on improving your online presence. I just should also mention that Michael is part of the FEMS Education Working Group, so has also been involved in the past and hopefully in the future in helping to put together our ideas for, you know, microbiology education. So thanks very much for coming here and representing our education group as well as your own university. But yes,
thank you so much.
No problem at all. Thanks a lot for joining me so early. And yeah, I hope we speak at another time.
Yes. Goodbye.
Bye.